Okay, so here we are. We'll go over the questions that I've had in discussions. So they've either been emailed to me or they've been messaged to me. So I put in bold the kind of the topic the questions are from and kind of paraphrased version of the question. So our first one is from genetics. So the student says, one question asked, DNA makes a good molecule for storing information because, and the answer is that because its bases can be joined together in any order like the letters of an alphabet can be strung to form a different word. So the student was confused by the idea that A's should pair with T's and C's should pair with G's, and so they didn't know how that worked together. So the idea is that when we do look at this base pairing, A and T, G and C, that's what's happening across a strand. So this T with that A, this A with that T, this C with that G, and so on. But what we are more interested in is the sequence that goes along the strand. So in this case, we've got TAC, TGC, CTA. All right, so you could have any one of those four bases in each one of those 9 spots there. So you don't have to have the sequence TAC, TGC, CTA. It could be all A's, it could be all T's, it could be all G's, it could be some other entirely different combination that we've got there. So it's this particular code that gives us the information that the DNA has. Okay, next question is a tall plant, big T is crossed with a short plant, big or little t. If the tall F1 plants are allowed to cross, self-pollinate, so the offspring. So in this case, what we're looking at, so is this incomplete dominant? So the F1 plants would be the offspring, right? So the tall plants, big T is crossed with a short plant, little t. All of their offspring here at one big T and one little t, so they're all tall, but they're also all heterozygous. And then if they self-pollinate, they're going to contribute one big T and or one little t, and so we can do a Punnett square to get the crosses. You can see 3/4 of the crosses are big T, have at least one big T, and then 1/4 of the crosses have little t. So in this case, because these tell us that they're all tall, we know that everybody that has at least one big T is also tall, and so these are short and these are tall. In incomplete dominance, we would end up with 25% being tall, these heterozygous individuals being a medium height, because incomplete dominance is kind of a blend of the two, and then these being short. So because they tell us here that these are all tall, then we know that these are tall, so we know that it's not incomplete dominance, it's normal Mendelian straight dominant recessive traits. Okay, the next one, the quiz said, which of the following does not provide evidence for evolution? The correct answer is natural variation within a species. I understood all the other answers provided, but I was confused by this as the workbook says that natural variation is the raw material. So in this case, yet natural variation is the raw material on which evolution works. But if we just go out and we sample a single population at any one time, we don't know how much that population is changing. In evolution, the definition of evolution is variation over many generations, variation over time, genetic change over time. And because we're just looking at variation at one time, we don't know what it's like over time, and so we can't say that evolution has occurred. It doesn't provide evidence for evolution. If we went back and sampled multiple times, and we did see that change, and then it would in fact be evidence for evolution. Okay, next one. Hi, Mr. Daddy. Hope you're doing well. I had a couple questions about the final exam. For a fall final, I found it very helpful to study out the previous tests and scantron answers and check. Since our distance learning test, I don't know what I got right or wrong in the recent test. I was wondering if for the final, you could release our past tests along with the right answers so I can make sure I'm studying the correct material. Because on some online distance tests, I do not know what I did wrong in each trial. Okay, so I do have a past test page on Canvas. It's the old test page. It's got the old test, but they're just the student versions. There's question only, no answers. So the closest I can recommend to getting the answers is to go back, use the workbook, textbook, online quizzes from earlier in the year, as well as these review quizzes that we've got, the Unit 4, Unit 5, Unit 6 review quizzes. They do give you the correct answers. That's really kind of the closest that I can do. I do want to say to do well, but we've also found there's also evidence that students who struggle a little bit to find the correct answer by going back and looking in books or trying to do some other information tend to remember those answers for longer. So that's really what we want. And also, I don't feel entirely comfortable just putting the corrected answers out there digitally. It feels a little weird to me. But if you do have specific questions about a message, without a question, message me or post it. Even better, post it on day 82 or 83 Canvas discussion, and I'll get to it there. And that gets kind of the end of the questions I've had. So if you have further questions, post them on the discussion page, and I will try to get to them. So, all right, thanks for listening.